Yeah.
So you see my screen well?
Yes. And
see my screen well, and my sound is okay?
Yeah, loud and clear. That's fine. And I think we're ready to start. Welcome to the Crowd Science Seminar. These seminars cover a broad area of human-AI collaboration to allow discussing past and future work, exchanging ideas, and establishing new collaborations. Today, Mohamed Amgad from the Northwestern University and Emory University talks about the application of crowdsourcing for annotating cell nuclei in breast cancers. That's a very intriguing topic. I have no idea how it works, but so let's focus on the talk. I'm happy to give the stage to the speaker.
Thank you so much. I really appreciate the kind introduction, and I'm honored to be here with you, everyone. My name is Mohamed Amgad. I'm a pre-doctoral fellow at Northwestern University and a PhD candidate at Emory University, Department of Computer Science. The work I'll be talking about today is about the application of crowdsourcing to a medical domain, and specifically for the detection and classification of nuclei, which are basically the fundamental elements that form tissues. This is me on the lower right. Since we're giving this virtually, some people might not know what I look like. And on the left is my PhD advisor, Dr. Lee Cooper. So let's dive to it. So why is this an important problem? So let me just give a very brief background, and I apologize if this is too basic for people with a biology background, but let's just to keep it for a broad audience. So what are we dealing with here? My work is mainly focused in breast cancer, and specifically, we try to characterize what is known as the tumor microenvironment. And the tumor microenvironment is basically looking at not just the cancer, but the area associated with the cancer, all the cells and all the associated tissue. And there's plenty of research that shows that not only is the cancer important for patient survival, but also the environment around which the cancer is operating. So the immune system that is fighting the cancer, the stroma or the surrounding tissue that sort of keeps the cancer in check from spreading, things like that. And the domain in which I am working is called pathology. And pathology is a medical domain in which we, if you have, let's say, a patient and they have breast cancer, they go and they undergo, a lumpectomy or some sort of operation to remove part of the cancer, or maybe they just do a biopsy to take a small piece of the cancer for diagnostic purposes. Now, that piece of tissue gets taken to the lab for someone to stain and look at it under the microscope to make a diagnosis or to make a prediction how long the patient will live, what kind of therapy they need, and so on. So this field is known as diagnostic pathology or diagnostic surgical pathology. And what you're seeing here in the center of the screen is one such sample. The things in pink are called stroma. They stain with a stain called eosin. And everything that you see that is sort of purple or violet is known as hematoxylin, and that kind of stains the more acidic elements of the cell, which tends to be the nucleus. So this is what a typical pathologist looks like day in and day out. They have a slide that contains a lot of tissue, and then they want to make a diagnosis based on the morphology. What do the tumors look like? Are they invasive? Are the cells dividing rapidly, which would indicate, of course, that the cancer is likely to spread, things like that. And recently, I mean, at least relatively recently, there's been this emerging domain known as computational pathology, which is the idea that you take these slides, which are normally physical slides that someone looks at under the microscope, and then you scan these using a very, very high resolution scanner. And then, then you can apply computational models to these and make a lot of these assessments that someone would do in a very subjective manner, do these assessments objectively. So if I look at these structures and I have to estimate what is the density of certain structures, let's say these TILs or tumor infiltrating lymphocytes, which is basically the immune system fighting back the cancer. If I look at this and someone asks me, what is the density of these things, and I have to make a ballpark estimate, this is not going to be very accurate. Whereas a computer, of course, can make these quantitative measurements much more objectively and in a repeatable manner. So this is kind of the goal, the fundamental goal of computational pathology. So again, the overall arching goals is to predict patient survival, know how long someone is going to live, if they're going to respond to certain chemotherapeutics, and of course, there's some science aspect. We want to find out if certain drugs work, some drugs don't work, and so on. So it's mostly on the prognosis and discovery aspect of pathology. And one specific marker that a lot of my work focuses on, and a lot of other people's work focuses on, because it's a very hot area of research at the moment, is something called tumor infiltrating lymphocytes. I briefly alluded to them. They're called TILs. They're these blue round cells that you see in the bottom here. And these are basically an indicator of your immune system going against the cancer. So when you see these things close to the tumor, so the tumor here is these elements that you see in a more violet with lots of nuclei. The nuclei are these roundish elements that contain the DNA, right? So if you see a lot of these cells close to the tumor, this means that your immune system is working and it's getting very close to the tumor to fight it back. So they're mostly considered a good indicator. If you see these in a patient, it means that they're responding to treatment, that they are likely to live longer, and so on. So there's a lot of interest in quantifying these things in an objective, quantitative fashion. So one of the difficulties from a computation, so now I mentioned the medical aspect of this, or at least briefly alluded to it. From a computational perspective, this is a kind of a difficult problem, because as you can see here in the various panels, there are so many variations and so many different patterns all within the same slide. So this is equivalent to, having a natural scene. If you imagine this is like a city, and having a lot of variation in the morphologies that you see. And of course, the exception here is that you don't even have ground truth data. Pathologists are busy. It's very difficult to produce ground truth, which makes this a peculiar problem. So it's, on the one hand, it's very important. On the other hand, we don't have data, and at the same time, there's no ground truth, right? Okay, so other challenges that we face with digital pathology has to do with scale. So these slides, when people think about ImageNet and MS-CoCo and other famous data sets, usually the images are fairly small, whereas in digital pathology, a typical slide is usually around 100K by 100K pixels. So it's closer to dealing with satellite imagery than it is to dealing with just small snapshots. And each slide contains a lot of nuclei, usually on the order of 500,000. And another problem that we face in this domain is just an extreme amount of variability. There's something called pre-analytical variability, because these are physical things, right? They're not just like CAT scans or X-rays where we take the image digitally and it goes directly to the system. But here we actually stain the slides. We take the tissue from the patient, we put some physical stain on it, and then we mount it on a slide and so on. And that is this causes an extreme amount of variability between different slides, different patients, different labs, and different disease types. Another problem that we face, that I also briefly alluded to it, is the fact that, when people talk about big data, it's true, we have a lot of data in digital pathology. Some centers have, hundreds of thousands of slides. The problem is that most of that data is unlabeled, and it's quite challenging to deal with unlabeled data in pathology. Because again, it's not about, for example, with a search engine, I know there are methods to use unlabeled data in clever ways to help you. digital pathology, there's some interesting methods that have been developed in recent, maybe 2 years or so, but for the most part, unlabeled data has very limited utility, and the amount of labeled data is basically almost close to zero. Everybody creates their own data, and then there are difficulty in sharing the data because of regulations related to patient privacy. So basically, there are very few public data sets. And another, of course, difficulty with this field is model confidence being very important, and interpretability being highly preferable, because you're dealing with people's lives and individual patients. So if a model tells you that it predicts with, let's say, 60% confidence that the patient is going to respond to this treatment, it really depends. That 60% confidence, is it good enough? Most likely it's not going to be. It has to be very high confidence, and it has to be able not only to give you the confidence of its predictions, but to tell you why it's making certain predictions, just because you're going to base a very big decision on that. So that sort of sets the stage for the difficulties in this field and the kind of problems that we're trying to solve with the algorithms that we are creating. So my specific work focuses on what are called concept bottleneck models. Now, I'm sure some of the people in the audience are familiar with this, being in the crowdsourcing and informatics domain. But I'll briefly allude to it anyway. So, typically in a lot of, not just digital pathology, but in explainable machine learning, usually saliency heat maps are given as like the be-all, end-all explanation. The model is looking here, therefore it's important. We're trying to sort of move away from that, because saliency heat maps, and there is quite a few papers that take this into some detail that I can maybe reference later, or people can email me if you want. But the problem with saliency heat maps is they just tell you where the model is looking. It doesn't tell you why it's making certain decisions, and it's quite subjective. And because interpretability is so important in the field that I'm working on, we are trying to move in the direction of concept bottlenecks. In other words, we use deep learning models, the power of deep learning, which you don't have to engineer any features. The model can ingest the raw image and produce a prediction using, of course, back propagation from one signal at the end. But instead of predicting the final output, like let's say you're looking at an X-ray and you want to predict if someone has arthritis, instead of predicting the final output right away, you predict intermediate concepts. Like, is there sclerosis? Are there bone spurs? Is there a narrow joint space? And because you're predicting these intermediate concepts, someone can go back and look at the, and of course, then later on, you can use some small regressor to predict the final thing. So when a model tells you, I think this is arthritis grade blah, or grade X, whatever that grade, you can immediately go back and see why the model says it's arthritis grade 2, for example. It says because it has a high probability of having sclerosis, of having bone spurs, and so on. So you can think of this as producing an embedding, but an embedding in which each member of that feature vector actually has a meaning that someone can take a look at and understand. Another advantage of using these concept bottleneck models is that they have a compressive capability. So what do I mean by that? Instead of having the model take the image, like the one on the left, and producing patient prognosis, the image is going to tell us what these individual elements are. Like, this is tumor. There's a tumor here. There's an infiltrating. So it takes the individual nuclei, the individual elements, and then from those, it makes the final prediction. So what this means is, if we would like to predict the outcome for a single patient, instead of having to feed in 100K by 100K slide, we can feed in very small tiles at a time, like one tile here, one tile here, one tile here, very small images, divided up into small squares, and then have these be represented by each one of these, instead of being a very large 2K by 2K image, each tile, it becomes a very small feature vector. And then these can be concatenated together to predict things at the level of a single patient. So it has a compressive capability. This is just saying that all the annotations that were produced are produced by software called the Digital Slide Archive, which you can refer in here. Okay, so all of this data has been produced, is piggybacking on a previous study that we did, where we gathered 20,000 region annotations. So what do I mean by that? So let me back up a little bit. What are we trying to do now that we established that we would like to use concept bottleneck models? We would like to detect the individual elements in the slide, and we would like to do it at 2 resolutions. The first is detecting homogeneous regions. So if you see here, everything in red is tumor, everything in cyan or blue is these immune elements fighting back the tumor, everything in yellow is dead tissue, right? So this is one level or one resolution of information. Another resolution of information is not the coherent regions, but the individual elements. So if you can think of this as detecting, let's say, a road with everything on it versus detecting the individual cars and individual people that are moving on the road, right? So one is a semantic segmentation problem, the other is an object detection problem. So we produced 2 studies in which we used crowdsourcing. So we used non-experts. We used the power of the crowd, non-experts, to produce data that would otherwise need a well-trained pathologist that needs years of training to produce. And the idea here is that some patterns are simple enough that a moderately trained crowd, so we're talking medical students or junior residents, but mostly medical students, people who have the incentive to do research and have and there's a lot of them, can produce, whereas otherwise we would not be able to produce this data because pathologists are very busy, their time is limited, and this task is boring, frankly. So this is basically what the focus of the study is. In a first iteration, we produced 20,000 region annotations for people from crowds, and we published that. And then in the second iteration, which is the paper that I'm presenting, briefly today, we produced 200,000 nuclei annotations. Both of the data sets are publicly available at grantchallenge.org, and there's plenty of people who signed up and started using the data. So this is very encouraging. So let me look at the bottom figure here. This sort of is just an outline of how we produce the bulk of the data. Essentially, the type of crowdsourcing that we are using here is what we term hierarchical crowdsourcing. The idea here is that you have a big crowd of people with minimal experience. So in this study, we had 25 of them. And then you have a much smaller crowd of people that are more senior, who can sort of supervise and give feedback and maybe produce some data for comparison. But the data, when I say 20,000 annotations or 200,000 annotation, I am talking about this part of the triangle, the bottom crowd. And of course, the triangle here is just sorting people by experience. So some of the data, the bulk of the data was produced in a single rater fashion. In other words, one slide is produced by one person, or one person can annotate multiple slides, but the same slide is only annotated by one person. That's the majority of the data. I'll say later how we control for the quality and so on, but for the most part, this is what the bulk of the data is generated. And then the people generate annotations, they ask questions on Slack. The pathologists or the more experienced crowd, they give feedback, and then finally, there are 2 coordinators that sort of make everything homogeneous and make sure it's publication ready and so on. So what do we use this data for? Again, I said one sample application is tumor infiltrating lymphocytes, detecting these blue, these circular elements that are fighting back the tumor. So I don't want to dwell on that too much. Let's jump to the actual crowdsourcing. So like I mentioned, we have tiers of people. And we annotated a bunch of classes. Now, in pathology in general, there's always a huge data imbalance problem. So the focus of this paper was not the uncommon or the at least the difficult or the visually difficult classes, because these need training and we don't have that in the crowd that we are using. So instead, we focus on the very three most predominant patterns that you would see in a high grade slide. So you see mostly tumor nuclei, these bluish, dense objects. You see fibroblasts, which are these faded cigar-like cells that produce the pinkish matrix that surrounds the tissue. And then lymphocytes, which are these, brownish, bluish elements that are fighting back the cancer. And in terms of data generation, we allowed people to generate a wide range of classes, but when it came to the modeling, we sort of generate superclasses, which is kind of common practice with crowdsourced data sets. One of the novel aspects in what we did was that because there's so many elements per each slide, there's, again, like 500,000 nuclei per slide. We're not annotating the entire slide, but still there's a lot of objects. And were we to do this fully manually, this would take forever. And we wanted to maximize the number of annotations. So we came up with a scheme to facilitate generating high volumes of data. And the way we did it is we generated computational suggestions for the people to see even before they annotate. And these suggestions were generated using models that do not require any data. So there are some existing non-deep learning models that are based on shape. And so the model that we use to generate these polygons that you're seeing here in panel A are basically things that look at darker elements that are roundish in shape. So they use very kind of low fidelity, low accuracy not machine learning, image processing to just produce very preliminary boundaries, some of which are going to be accurate, some of which are going to be complete rubbish. But they produce these, and then we inherit the class from the region. So if you remember, I said earlier, if you see in panel B here, that we generate an initial data set where people would circle the low resolution regions. So here we make the assumption that every object in a particular region inherits the class from that region. So for example, if you see anything, in the, let's say, self-driving cars context, this would be like assuming that anything that you detect on a road is a car, right? Until proven otherwise. And the idea here is that people would get suggestions. Again, you have a crowd that is not trained to do this kind of thing. They just receive a very, low, not a very, just a brief training. But you want to sort of guide them, or at least help them get a lot of data quickly, and also help them discern what the class might be. So this is exactly what we did, and this is what the person sees. They had access to both the high resolution data as well as the low resolution data that another crowd generated in the previous iteration. And then when they see a boundary that they like, they place a dot, right? And if they see something that they don't like, they just place a bounding box. So you can immediately see that this gets us a new type of data where the same field contains a mixture of segmentations, right, which were not generated manually, they were just clicked as being accurate, as well as bounding boxes. And the idea here is generating segmentations takes a long time, and we would like to maximize the number of data that we get. So what you see here in the bottom right is the type of data that we get, which is a mixture of bounding boxes and segmentations. I already explained this. So we have a heuristic image processing algorithm that generates these segmentations, and then we have the object inherit the class from the region in which they are. One new aspect that we came up with is using the object detection and segmentation framework Maskrcnn as a function approximator to improve the suggestions that people see. So essentially, what you're seeing here is an actual actual image from the data set. So because these heuristic algorithms are weak, here you can see that they missed a lot of the tumor nuclei in the lower right corner, just because they are lower contrast nuclei. But then when you train on the entire data set, your Maskrcnn here acts as a function approximator. So here we're not using it for inferring new data. We're just using it. This is similar to fitting a regression line through noisy data and then using that regressed line as your suggestion. So this is exactly what we did, and this gave us better suggestions that people looked at when they were making their final assessment for the annotation. So I already explained the panel on the top here, which is the single rater data set. Another way that we ensured quality is using a lot of control experiments. So in our control experiments, we would have the same slide be annotated by everybody, right? Or at least, as many people as possible, both the senior people as well as the junior or the non-experienced people. And then we would use, truth inference to infer what the experienced crowd thinks the annotation is and what the non-experienced crowd thinks the truth is, right? So we would do some sort of clustering and expectation maximization and other truth inference methods to just figure out what the different people think the various nuclei are, and then compare them to each other just to see if one person is more accurate than the other, if the crowd is doing poorly, and so on. Another set of control experiments that we did was looking at whether or not these suggestions bias people in one way or the other. So we asked the same person to annotate the same area without looking at suggestions first, and then there's they wait, and then later on they look at the same image and then they see the suggestions without the function approximation, so using these poor low fidelity image processing, and then they wait a little bit and then you look at the same image with the refined suggestions, the Maskrcnn function approximation. That way we can sort of compare the same person, the same image, and we can single out the impact of the imaging suggestions, just to see if these bias the data in one way or the other. Okay, so I'm going to skip the truth inference because most of you will be familiar with it. It's just a simple clustering of the spatial locations where everybody placed their annotations, and then using expectation maximization to infer both the participant reliability as well as the final class. So the end result would be for each nucleus what we think the actual class is given everybody's annotation, as well as how good is every single participant for every single class. So there was no novelty there. We just use an existing method. Okay, so let's look at some accuracy results. What you're seeing here in panel A, for example, is this is a scatter plot of how individual people are doing relative to the inferred truth. So the inferred truth is we have pathologists annotate an image and we infer what the actual nucleus location, and actually the locations are fixed. So we infer the class, right? And here we look at the detection accuracy. So given a particular location, everybody is assigned a label. They detect whether they detected that nucleus or they didn't, and then we can get the accuracy relative to the actual truth. And then you can see here that the people who tend to do best in terms of detecting nuclei are the junior pathologists. Basically, and our hypothesis here is that junior pathologists, or people who are just training to be pathologists, have more time. So they annotate more nuclei, even if something is faded, or they just take the time to annotate everything. But everybody seems to be doing well in terms of detection, give or take, with the exception of 3 non-pathologists here who missed a lot of nuclei. In panel B, we're showing the classification accuracy, both when you group things by superclass and when you don't group things by superclass. And I guess the fundamental thing to take away from this slide here really is that people are accurate as long as we look at the micro average. So as long as we don't care about the uncommon classes. The moment you start caring for the uncommon classes, things like faded fibroblasts in the background, or things that are not common, this is where you see a dip in the accuracy. And this is not unexpected. Your crowd is not really well trained, right? This is the whole point of the study, and this is a difficult problem. And that's why you want to see high micro average accuracy, because that's really the majority of your data set. And then later on, you can supplement your data set, of course. If you want to use this in a real setting for, if you care about these rare classes, then you can have pathologists, people who are well trained, supplemented with these uncommon classes. Another thing I want you to take a look at is panel E here. I'm going to skip some data if it's less relevant to the talk, but panel E here I think is interesting. We wanted to ask the question, exactly how many people should annotate the same region in order for their data to be trustworthy, right? So this is an interesting question, because in the real setting, you want to know how many people you want to recruit. And or if you're recruiting a constant number of people and you're letting people annotate things at their leisure, which is most likely going to be the case, if you have an image that is annotated by 3 people or 5 people or 7 people or up to however many, up to 15 in this case, how many people do you actually need in order for the data to be trustworthy, given that the people themselves are not individually trustworthy? And the idea here, we just used a simple simulation where we have a matrix. The actual data is we have 15 people who annotated every single image for this subset of data. And then we just decimate. We randomly remove annotations to simulate missing data. So, in one iteration, we'd have only 3 people annotate the same slide, and another we'd have 5 people annotate the same slide. We kept the number of people, the number of unique people constant, just to control for the variation in it acts as a control. Keeping the total number of people constant for this simulation acts as a control. But then we decimated the number of unique people per image. And then what we see is that overall, even with 3 people, you can get above 0.9 AUC, but really it's class dependent. So if you care about, let's say stromal cells, you need 6 or more people per image. If you only care about, tumor and the elements that fight the tumor, then you need 3 people per image. So the main conclusion here is that it is class dependent, and that for the uncommon classes, you need more people, which of course makes sense, because the individual people are unreliable. So the more of them you get, the more likely that their combined truth will be trustworthy. And of course, here again, the assumption is that these patterns are these people at least have some knowledge of these patterns, which is usually the case for the common things. They're medical students. They have at least some pathology training. Here we investigated the effects of algorithmic suggestions. This is the control experiment that I talked about. And if you look for a moment in panel A here, all of these images are from the same person, right? So if you look at the top image here, this is the same person annotating the image without seeing suggestions, and then with seeing suggestions. And then whenever you see a dot, this basically means that this boundary was not created manually. This was an automatic boundary that the person found to be accurate. So you can see that when people see suggestions, they can generate quite a few boundaries for free, right? They didn't have to spend the time to move the mouse around the nucleus boundary and so on. So this is kind of the main thing to see here, is as you move to the right, there are more dots, which means that the suggestions help in getting data for free. This is the same. Panel B and C say exactly the same thing. So what you're seeing here, let's focus on the evaluation data set, which is our main data set, our main multi-rater data set. What you are seeing here is that when people see suggestions, they can click on up to 40% of the nuclei, which means that up to 40% of the objects, they will have boundaries for free without people actually generating boundaries, which is pretty good. If you have, if you're generating, let's say 100,000 boundaries and 40,000 of those are generated for free, that's a pretty good yield, for your annotations. What you're seeing in panel C here is that the segmentations of the boundaries that people click are that basically people click on a boundary when it is more accurate. So we have a small data set that we generated where we have manual boundaries, and we're comparing this to them. So you know, let me put this in other words. What is the accuracy of a boundary that someone clicks compared to the same boundary if someone creates it manually, right? And then what you're seeing here is if someone clicks on a boundary, on an algorithmic boundary, it's significantly more accurate than an algorithmic boundary that the person does not click. This is what this is saying here. And in the bottom panels D and E, they show something pretty interesting. Here we are asking, are people swayed by looking at the classification of the nucleus? So in other words, we're asking if someone looks at a particular nucleus and the algorithm is telling them that this is probably tumor, right? So let me go back up on here. So you see that the suggestion, the algorithm is telling the person, this is probably blue, right? This is probably a lymphocyte. This is probably red. This is probably tumor, right? So do these suggestions bias people in one way or the other? That's what we're asking. And if they do they bias them in a good way? Do they make them create better data? Or do they bias them in a bad way, making them generate worse data because, of course, you know, they're sort of kind of obscuring the object, or they may be, right, they're biasing their opinion. So this is what that figure was showing. And we break it down by people who are experienced and people who are not experienced. And here are the main conclusions. Both the pathologists and the non-pathologists are swayed to some extent by suggestions, but they still have substantial agreement with themselves. So here we're looking at self-agreement. The same person, do they agree with themselves or not? So there's substantial agreement for both pathologists and non-pathologists, but the agreement is quite remarkably higher for pathologists. So the pathologists are going to be consistent with themselves whether or not they see a suggestion. So they know what the object looks like. It doesn't matter what the computer tells them. The non-pathologists are actually more impressionable. So you can see that there's a dip here. If you're a non-pathologist and you see a suggestion, you are likely to change your opinion. But panel E here shows that you change your opinion for the better. So if you look at the solid ROC curve compared to the dashed one, the dashed one is the unbiased. This is your accuracy as an inexperienced person when you don't see suggestion, whereas the solid line is your accuracy when you see a suggestion. So what we are saying here is that the non-pathologists are kind of more easily swayed by seeing suggestion, but they're swayed in a positive direction. They produce better data when they see the suggestions. And of course, I know that this is dependent on how good the suggestions are, right? So this is a given. We are here making, in this study, we are giving the computational suggestions based on a previous study that people generated on bulk. Basically, they just annotate the entire area as like, this is tumor. And so this is basically saying that if we make the assumption that every person on every object on a road is a car until proven otherwise, this is the equivalent here, that this actually helps people annotating data when they see the suggestion that this object is probably a car. Hopefully that makes sense. So just to take this one step further, we did some modeling with the data. So just one unique aspect of this data set is that the size of the object is much smaller than you would in a traditional data set like COCO or ImageNet. Or so the NuCLS data sets, they tend to have a lot of small objects, very small objects that and too many of them. You know, on average, there's 34 objects per image, and they tend to only occupy like 1% of the image. So it's a fundamentally different from a lot of other data sets out there. To make a long story short, this required us to make modifications to Maskrcnn in order for it to work well with this kind of data. Most importantly, when we are calculating the loss, we ignore the mask loss when we have a bounding box. So this is basically a very simple modification to deal with the fact that our crowdsourcing annotation method generates a mixture of bounding boxes and segmentations. And another thing we did is we relied on the fact that all of the objects in the type of data that we're collecting, all of the objects have a constant shape or similar shape, which is ovoid in nature, which allowed us to use something called class agnostic non-max suppression. So if you're familiar with Maskrcnn, this is what we did. If you're not, please ignore what I just said. Here, just showing some pretty figures of the the accuracy of showing both the accuracy of the models that we generate using this data, as well as the fundamental ambiguity of the task. So here, whenever you see an arrow, these yellow arrows are errors, and you can see sometimes you see the errors in the truth side, right, in the bottom panel. So here, the error is that the person just did not annotate the individual nucleus, whereas the model detected it. Sometimes the error is that the person thinks that this is an ambiguous nucleus and they don't want to give it a class, right? But of course, sometimes you will see the problem with the algorithm. Like here, the algorithm misclassifying things, or honestly, sometimes it's hard to tell. The case is inherently ambiguous, but in other cases, it's a true proper problematic misclassification. So in the bottom right here, there's a lot of mistakes that the algorithm is making, and it's mainly because this is for a rare type of nucleus that basically non-pathologists, these non-experts, are just not equipped to annotate. So we expect these kinds of errors to pop every now and then. Another sort of here I'm sort of describing two papers really. The modeling is another paper, but I thought it was interesting because we came up with this technique that we call detail or decision tree approximation of learned embeddings. And since the whole sort of pattern that the theme of our work is explainability, one thing we came up with is we wanted to move away from saliency. So one thing we did is detect these individual elements, the concept bottleneck models that I described at the beginning. Another thing that we did is we wanted our models to be able to not only find these individual nuclei, but to even explain why it thinks this individual nucleus is tumor, this individual nucleus is fibroblast, like why this one is assigned the red color, why this one is assigned the green, and so on. And what we came up with is a really simple scheme, whereas we fit a decision tree, but instead of having the decision tree explain, instead of having our decision tree fit to the truth, we have the decision tree fit to the prediction. So we use a regression tree, we fit it to the embedding. So this is why it's called learned embedding. So our model produces an embedding, our trained Maskrcnn. We train a decision tree to produce the same embedding using features that we can understand. So this is our model, this is our nucleus model, this top here in panel A, and then we train a parallel model that uses features that a pathologist or anyone who understands data can understand, like what's the shape of the nucleus, what's the staining, where are the edges, to produce the same embedding, right? Which means that when we look at the decision tree, this would be a human explainable explanation that best approximates what our black box model is doing. So if we look at let's look at the three common patterns. If the major axis length of the nucleus is high, above a certain threshold, then you go to the right. And if also the minor axis threshold is high, then you go to the right. So if both the major and the minor axis length is high of that object, chances are it's tumor, right? If, on the other hand, it has a small major axis length and a very high circularity, chances are it's this lymphocyte, this element that is fighting the tumor. And then if otherwise, if the major axis length is high, but the minor axis length is small, which means that it is cigar-shaped, then chances are it's a fibroblast. So this is distinguishing these big globular nuclei versus the cigar-shaped nuclei versus the small around nuclei. And then we know that our model is using something like this when it's making its decision, because the decision tree was fit to the prediction, not to the truth. So that's it. I just want to thank everyone who contributed to this data, the big crowd. I know for some of the crowdsourcing work that you guys do, this is not as big of a crowd, but in medical applications, it's kind of a big crowd. So I want to thank each and every one of them, many of whom went on to actually become pathologists. And of course, I want to acknowledge the funders who helped this study. Thank you very much, and I'll take your questions.
Yeah, thank you very much for the great talk. Questions, please. Do we have questions in the chat? Nope. And questions from the audience? I think I can start the discussion. Yeah, Daria raised the hand. Could you please come in and ask the question?
Yes. Hi, Mohammed. Thank you very much for the talk. It was very interesting and engaging. I just have a very small question. So you said that you were asking for help from non-pathologists, like those students with minimal experience. However, they were still medical students nonetheless.
Yes.
Do you believe you can do and complete like other similar crowdsourcing tasks using just an open crowdsourcing platform where you would have to deal with people with no medical background whatsoever? And do you think this would be possible? And if it would be possible, how? Thanks.
Yeah. Well, thank you for the compliment, and I'm very glad to be here. I think it is possible in a limited setting. So the short answer is yes. The long answer is it will have to have a very well-structured training that people have to go through and pass before they are allowed to annotate. And just like with the medical students, they would be limited to these very common visually distinctive patterns that don't rely on architecture. So there's a difference between patterns that rely on architecture, things that where you have to integrate a lot of information and know a lot of clinical associations to make. These even medical students cannot make, most likely. But as long as we are talking about things that rely on texture and slam dunk object recognition, yes, I'm optimistic that a non-trained crowd, non-medical crowd can do it, as long as there is a test at the beginning and a small training. For example, medical students, let me give you a concrete explanation here. Medical student, you don't have to tell them what tumor is, what a lymphocyte is, what is a nucleus, things like that. A medical crowd would because they, medical students have some basic knowledge of pathology. What a non-medical crowd would have to have is a module that is probably, something like 2 or 3 hours of training, followed by a short test where they just get these fundamentals out of the way so that the most obvious mistakes are not made. I also suspect that if you do a simulation with a non-medical crowd, the number of unique people per image that you would need for it to be trustworthy will be higher. So these would be your constraints.
Okay, well, thank you for your optimistic, opinion, because a lot of people really, don't necessarily believe that people with the zero medical background can even do any kind of, object segmentations. And what we've seen in our experience, exactly what you're saying, you just need to invest a lot in the effort into trainings, and usually you should even pay higher to those people, just because it takes much longer time to get to know the tasks and stuff. Cool.
Yeah,
thank you so much.
I would even say that's the kind of the ideal situation, because medical students are great, but the numbers are still limited. And so there's a question of incentives here. The people in our study, the incentive was recommendation letters and publication co-authorship, things like that. In the real setting, when you want to generate more and more data, financially, that becomes really the main thing. And medical students are, some of them may be motivated financially, but that's less likely. And that's when you actually do need the non-medical crowd.
Yep. Thank you very much for answering. Thanks.
Yeah, thank you for the question and the discussion. Do we have more questions from the audience? You have a rare chance to ask something related to, medicine. And in the chat, there is a question: how fast error rate of non-specialists went down in number of annotated tasks?
That's a very good question. So we didn't actually systematically test that question. And the reason is it has to do with just how the platform that we were annotated with, it's a platform for annotation, but not for crowdsourcing specifically. I suspect if we used a crowdsourcing focused platform, something like Crowdflower, and again, and the reason we didn't use something like Crowdflower or Tukola or other crowdsourcing platform is that these whole slide images, they are extremely large, so you need some sort of it's like specialized software to view and pan to the slide and so on, right? And that is not something we had access to. If we did, we absolutely would use a crowdsourcing focused platform, because then we can answer questions like that, right? We can track when the person started annotating, and then we would track how, which annotations were created first, which annotations were created last, and then we can backtrack and see their accuracy change over time, or maybe even discard things that were generated first and only keep the ones that were generated last. Things like that would definitely is something that we do look forward to in future iterations of crowdsourcing platforms that can support panning whole slide images. That's really the main limiting factor there.
And there is one more question in the chat. You said that more experienced participants could also teach the non-experts, right?
Yes, they can. Yes, they can. There are modules, and the thing is, medical students, they are trained, but usually in medical school, medical students get trained in what is called pathophysiology. So it's not the pattern recognition aspect that they get really a lot of training on. It's mostly the diseases and how these patterns tie to, diseases and so on. Which, to put this in other words, even medical students don't have a lot of training on the pattern recognition aspect of pathology. So as much as it seems that this is really far away from, this study, from dealing with a non-expert crowd, I would say we were pretty close. I would suspect that, with the exception of that initial training, the results wouldn't be too different, just because medical students are close to a non-trained crowds. A lot of people, when they go to pathology residency, it's like they're training on a completely different field, actually, in terms of the pattern recognition, not in terms of the disease associations.
And
so yeah, they would be able to be trained, because if the medical students could give us this data, the non-experts or non-medical audience should be equally capable of producing something like this.
And as a follow-up question, there is a diagram on this slide that shows the hierarchy of the annotators, right? So you have a very few number of pathologists that can actually guide the whole annotation process, but they are too busy to cover every possible case. And you have literally a crowd of non-expert annotators who can provide these labels. So you build the pyramid of the annotation expertise. My question is, what was the incentive for being a part of this study? Because as far as I see, you used Slack, and it's probably something based on the volunteer effort, which is perfectly fine. But what was the initial premise? How did you invite people, and why they are there?
Yeah, that's a very good question. So in these studies, the incentive was purely academic. You annotate your share of the annotations with us. You are a co-author, assuming, of course, you look at the paper and provide other intellectual contributions. And if a lot of these medical students, they wanted to go through to residency, so you get a recommendation letter stating what you did. So it was purely academic and purely career focused. I suspect that, you know, replicating this study would require one of two things. One is, if you really want to do this systematically, that this is done through a medical school or through some, you know, something more systematic. That way you can really replicate this. Or again, you completely branch out and introduce financial incentives, which is would be my preferred recommendation if the funding were available, actually.
Yeah, and, thank you. And when it comes to the neural network, so currently I see you have a very sophisticated pipeline that actually uses some Slack annotation with the crowd of volunteers, and also you have the machine learning part that takes the obtained image segmentations. Please correct me if I am wrong. And you predict the classes, but I really loved the slide that mentioned that the class distribution in your data set is skewed. You have some of the classes are, you know, rarely underrepresented, some are overrepresented, and you managed to, you know, address this problem as far as I see to some extent. And I see that you try to use, you know, class grouping, and but how it worked entirely. So you have these classes that are underrepresented, and what do you do to actually handle this classification?
Yeah, so this is actually the main problem that we face with this kind of analysis. You're absolutely right. Our main issue and difficulty in dealing with this data, even now when I use it for downstream analysis and other work I do, is how do we group things and deal with the skewness in the data. So let me tell you the ideal situation if I had unlimited, not unlimited, but like more time and more access to financial resources and so on. What I would do is I would have a field I would ask the non-pathologist only to annotate these big things, these, you know, big pattern distinctive classes, these, you know, the red, the green, and the blue. And then I would do a second round where I have the pathologist go through the exact same fields, but only looking for these uncommon ones. That way you can sort of balance, you know, you make sure that the pathologists take the unbalanced. In our data set, of course, we don't have access to this kind of thing, partly because the pathologists are busy and so on. So what we did is we just thought, from a pattern recognition perspective, which of these things, from both a pattern recognition and a medical perspective, which of these things are okay to misclassify or to confuse with each other. That was the basis of our grouping. So tumors and mitotic figures, they actually look visually quite distinctive many times, but really it doesn't matter. And most of the people will annotate a mitotic figure as a tumor, and it doesn't matter if you misclassify these for most problems. Sometimes for breast cancer, mitotic figures are very important, of course, but you know, for the most part, if you're doing large scale analysis, it doesn't matter. So this is how we came up with this kind of hierarchy, and it served us well. Actually, dealing without the hierarchy, first of all, you lose a lot of data because these uncommon classes, they're uncommon individually, but when you add their numbers, that's a, you know, small, significant chunk. But also, you know, their misclassification doesn't really matter for the most part. So why not? That was the reasoning that we gave.
But when it comes to the machine learning machinery, what happens then? So in your data set, you have a segment, and do you attach the superclass or the raw class to the?
Oh, I see what you're saying. So what we ended up doing is, first of all, the raw class was not used for anything, just because it you know, there are some things that have like 4 annotations in the entire data set, and then when you split, you know, train test, you know, this gets completely lost. So what we did instead is we did some reasonable main class set. This is what we used for training, but then when we looked at the accuracy and we actually try to assess the performance, we group things. So anything that the algorithm predicts as a mitotic figure or tumor, they're both considered tumor. Stroma or macrophage, both considered stroma, and so on. And what we found in our preliminary experiments is that actually training models on classes gives us better superclass performance. I guess now it's a very non-scientific way to put this, but the idea here is that when you place things that look different into different buckets or different parts of the embedding space, that sort of helps things when your model is training better than trying to learn to lump things that actually do look different. Like if you look at the tumor here in the red and some mitotic figures in yellow, you can see that these kind of look different. And we found that even if we don't care about differentiating these when we're doing the inference and looking at the accuracy, when training the model, we want to put them into different buckets. And I'm not sure if I included that slide here. I didn't. It's in the paper. What we show is that actually placing things that look different into different buckets during training gives us better performance when we look at the superclass accuracy. Kind of peculiar, but just turned out to be the case.
Got it. Yeah, and just a very stupid question, but scientometric. How much time did you spend to finish the study?
Well, it took longer than I would like to admit. It took, I'd say, 1.5 years, but there were other things happening in parallel and so on. So it's hard to say the exact time, but yeah, I would say 1.5 years, given the recruitment and writing the paper and dealing with stupid mistakes. It's an academic study, so I know in an industry setting probably will also take the same time, just because of all the extra safety things that you guys will probably take into account related to quality and so on.
Very good, very good. And when it comes to the industry research, we have a similar situation. We also have mistakes that we make. We are still humans, and maybe it's not that expected, maybe, but we still do them. Yeah.